Hello guys, this is Akshay over here and welcome to my another video for the KCT and NEET preparation discussion. And in this particular discussion, we will be mainly looking into a completely new chapter, that is body fluids and circulation. Again, a small part of this particular chapter. So let's get started. So blood. So blood is mainly a special connective tissue which is mainly made up of a fluid matrix, and it also contains the plasma, that is the fluid part of the blood, and also you find the formed elements in the sense mainly the cellular components of the blood. So plasma makes about 55% of the blood's composition, and around 90 to 92% of plasma is made up of water, and you find that it is having only 6 to 8% of proteins. And what kind of proteins you find in the case of plasma? That is mainly the fibrinogens, globulins, and albumin. Fibrinogens are mainly involved in clotting of blood, but whereas globulins are mainly involved in immune reactions or defense mechanisms, and albumins are mainly involved in maintaining the osmotic pressure. You also find ions such as the sodium, calcium, magnesium, bicarbonate ion, and also chlorine. So if you remove the clotting factors from plasma, it is termed as serum. So the next one we can look into is the formed elements. So you mainly find RBC, leukocytes, and platelets. All of these three make up the 45% of the blood. So looking into the RBC, the normal number of RBC in the case of healthy human is around 5 to 5.5 millions per mm cube, and it is mainly formed in the bone marrow. It is having a red color, and it is mainly because of the hemoglobin, and it is having heme group along with iron. So what should be the normal quantity of hemoglobin in a healthy person? It should be around 12 to 16 gram of hemoglobin in the blood. The normal lifespan of the RBC is around 120 days, and it is mainly destroyed in a special organ called as the spleen. So moving on, the next one we can look into the is WBC. The number of WBC in a healthy human is around 6,000 to 8,000 mm cube, and you find mainly two types of WBC, that is agranulocytes and one more is the granulocytes. So now granulocytes are of again three types. The first one is the neutrophil, eosinophil, and basophils. So the neutrophils are around 60 to 65% of the WBC population, and they are mainly phagocytic in nature. Eosinophils are around 2 to 3%, and they are mainly involved in allergic reactions. But whereas basophils are the least in number, and the population is around 0.5 to 1%. They are mainly involved in secretion of histamine, serotonin, heparin, and they play mainly important role in the inflammatory reactions. So moving on, we are having A granulocytes, which you find mainly of two types. The first one is the lymphocyte, which is around 20 to 25% in the population, and one more we are having is the monocytes. So in the case of lymphocytes, you find again two types, that is the T cell, and one more is the B cell. Remember, T cell is mainly involved in cell mediated immune responses, but whereas B cell is mainly involved in the humoral mediated immunity or responses. So the next one we are having is the monocytes, that is mainly 6 to 8% of the population, and they are mainly phagocytic in nature. Remember, from the monocytes you also get one more type of cell, that is the macrophages. So the last we are having is the platelets, which are mainly the cell fragments derived from the megakaryocytes, and the normal number in the healthy human is around 150,000 to 350,000 mm cube. So moving on, the blood group system in the case of humans. You can see that there are four different blood groups in the case of humans. The first one is the A, B, AB, and O. The speciality is that the person with the A blood group is having an antigen of A on the RBC surface, but whereas the plasma contains the antibody B. So when it comes to blood donation, people with the blood group A and O are the people who can give blood to the person with the blood group A, right? And next is the blood group B. In the person who is having a B blood group, you can see that the antigen on the RBC surface is again only B, and the antibodies in the plasma are antibody A. So people with the blood group B and O can donate the blood group to the people with the blood group B. And next one we are having is the blood group AB, where you can see that antigens on the RBC are both A and B. Both the antigens A and B are present on the surface of the RBC, but whereas you don't find any kind of antibodies in the plasma. And the people who can donate the blood group to the AB people are AB, A, B, and O. Hence, we refer AB blood group as a universal acceptor. Moving on, we can see the last blood group is the O, and you don't find any kind of antigens on the RBC surface, but whereas in the case of antibodies in the plasma, you find both the antibodies, that is A and B. And the people who can donate blood to the O people is that only O. So the person who is having O blood group can get the blood from the people who are having O blood group only. But you can see that people with O blood group can give blood to anyone. Okay, you can see over here, right? Hence we refer O blood group as a universal donor, AB as universal acceptor, O as universal donor. So now here you see an interesting hemolytic disease called as the erythroblastosis fetalis, which mainly arises because of complication in the Rh antigen. So Rh antigen is again present on the RBC only. So remember, the person who is having Rh antigen, we refer them as positive, and the person who is not having Rh antigen, we refer them negative. For example, if the person's blood group is A positive, it means that the person's blood group is A, and the plus sign indicates the person is having Rh antigen, hence A positive. If it is A negative, the person's blood group is A, and the negative sign indicates the person is not having the Rh antigen, right? So when a woman with Rh negative marries an Rh positive person, during her pregnancy, the fetus that is developing is actually Rh positive. So what happens is that as the mother is Rh negative, but the developing fetus is Rh positive, so when there is an exposure of fetus's blood to the mother's blood, immediately there is an immune response and antibodies are actually produced against the fetus's blood because the fetus blood is having Rh antigen, and this antigen is considered as a foreign body. So what happens is that the first pregnancy is completely normal because the time duration that is required to prepare the antibodies in the first pregnancy is more of time, and in this particular period of time she delivers the first baby in a most healthy way, so there's no problem. But the problem arises in the subsequent pregnancies where the mother's blood has been already exposed to the fetus's blood, that is Rh positive, in the first pregnancy itself, and in the subsequent pregnancy, if there is any accidental exposure of the fetus blood to the mother's blood, immediately the mother's antibody enters or crosses the placenta and enters into the fetus and mainly ruptures the RBCs of the fetus. And this is actually an hemolytic disease. Hemolytic means rupturing of RBCs. And this rupturing of RBC of the fetus is called as the erythroblastosis fetalis. So moving on, we can look into the blood clotting mechanism. So you can see that when there is any particular wound, there is a formation of clot, right? So this clot formation is mainly because of a special protein present in the blood called as the fibrin. So now the fibrin is mainly involved in the formation of fibrin thread and also the clot formation. But however, you see that the fibrin is actually present in the form of inactivated form called as the fibrinogen. So activating the fibrinogen to fibrin is because of an enzyme called as the thrombin, but you also see that the thrombin is again present in the form of an inactivated form called as the prothrombin. So to convert the prothrombin to thrombin, you need an enzyme called as the thrombokinase. Activation of thrombokinase is again under the control of calcium ions and certain factors derived from the platelet. So whenever there is a wound or cut that results in the blood oozing, you can see that calcium and factors from the platelet activate the thrombokinase, and thrombokinase converts the prothrombin to thrombin, and thrombin regulates the reaction of converting the fibrinogen to fibrin, and when finally the activated fibrin is present in the blood, it mainly forms the fibrin thread and the clot is formed. Finally, the bleeding is stopped. So this is the blood clotting mechanism. So the next one we can look into here is the lymph. So lymph, what is basically lymph? You can see that the water content from the blood actually moves into the spaces or passes into the spaces between the cells of the tissues, and this space between the cells of the tissue is called as the interstitium, and the liquid which is present in this particular space is called as the interstitial fluid. And this interstitial fluid is mainly required for exchange of gases between the blood and the tissues. So this particular interstitial fluid is collected by a network of vessels called as the lymphatic system, and now this fluid which is present in the lymphatic vessels is termed as the lymph. So basically lymph is a colorless fluid which is mainly containing some specialized lymphocytes that play an important role in the immune responses. So you should also remember from our digestion chapter that fat absorption is mainly mediated through the lymph. So finally, we can look into the circulatory pathways. So you know that the circulatory system may be of two types. The first one is called as the closed circulatory system. That is, if the blood is flowing in the blood vessel, it is called as the closed circulatory system. But if the blood is directly flowing into the body cavity, it is called as the open circulatory system. You see this kind of open circulatory system in the case of arthropods, but whereas in the case of closed circulatory system, you find in the case of vertebrates, or one more example we are having is the annelids, right? So the circulatory pathways are again of three types. The first one we find the single circulatory system, single circulatory pathway, incomplete double circulatory pathway, and finally we are having is the complete double circulatory pathway. So the single circulatory pathway is seen in the case of fishes, because the heart in the case of fishes are mainly made up of two chambers, that is a single auricle and single ventricle. So the heart actually pumps the deoxygenated blood to the gills, where it gets oxygenated, and this oxygenated blood is directly to the body tissues, and after supplying to the body tissues, this blood again comes back to the heart in the form of deoxygenated blood. So this cycle is called as the single circulatory pathway. But again, in the case of incomplete double circulatory pathway, it is seen in the case of amphibians and reptilians, because you find that the heart in the case of amphibians and reptiles are mainly three-chambered, that is two auricles and one ventricle. So the right auricle is mainly involved in receiving the deoxygenated blood, but whereas the left auricle is mainly involved in receiving the oxygenated blood. So both the oxygenated and deoxygenated blood enters into a single ventricle cavity. As a result, there is mixing of blood, and this completely mixed blood is pumped to the different parts of the body. So this kind of circulatory pattern is called as the incomplete double circulatory pathway. And finally, we find the complete double circulatory pathway, which is seen in the case of aves and mammals. So we all know the four-chambered heart, that is two auricles and two ventricles, is seen in the case of aves and mammals, right? So the ventricles are completely separated in the case of aves and mammals. Right auricle receives the deoxygenated blood, and the left auricle receives the oxygenated blood, and the deoxygenated blood from the right auricle goes into the right ventricle, and from the right ventricle this blood is pumped to the lungs for oxygenation, and this oxygenated blood returns back into the left auricle, and this oxygenated blood from the left auricle enters into the left ventricle, and from left ventricle through the aorta it is pumped into the different body tissues to supply the oxygen. As you can see that there is nowhere mixing of blood. Hence, this kind of circulatory pathway is called as the complete double circulation. So we will stop our discussion over here. So if you found this particular tutorial very much helpful, then please do like the video and share the video, and finally subscribe to this particular channel. Thank you.